Good afternoon, everybody. My name is Matthew Phillips, neurologist, and today I'm going to first start by just thanking you for attending my talk, and I'm also going to thank the conference organizers for MDS ANZ 2022. So with that being said, I'm just going to get my screen up here. Okay, so today's talk is on metabolic strategies in Parkinson's and Huntington's, and I hope by the end of this talk you understand what I mean when I say metabolic strategy. So in neurology, we often talk about location and pathology. As clinical neurologists, we are classically trained over a period of many years to so-called localize the lesion by trying to find the location and pathology of a lesion in the body and then see what we can do about it. This is undoubtedly diagnostically useful. However, the conceptualization of a focal pathology in an otherwise healthy body is actually, if you think about it, somewhat artificial. Particularly in Parkinson's and Huntington's, which are widespread disorders involving many structures, because basically the rest of the body may not be so healthy. So let's look at location first. We often localize Parkinson's and Huntington's to this deep set of brain structures called the basal ganglia. And this is a commonly used diagram to show the sort of circuit connections between the various structures of the basal ganglia. The names aren't too important. I guess basically current thinking is that the basal ganglia are involved in selection and switching of behaviors, which you can think of as being neocortical representations, so lying within the neocortex, that thin envelope of neurons around the outer edge of the brain. And the basal ganglia are thought to function basically as a competition between two pathways. So the first pathway is the green one on the right there, with the green box, the indirect pathway, which is thought to basically inhibit the thalamus and cortex and result in sort of a general inhibition of behaviors and movements. And Parkinson's is possibly related to an excess of activity in that pathway. Conversely, we have another pathway, the direct pathway, the one outlined by purple, and that stimulates the thalamus. It actually disinhibits the thalamus and the neocortex to allow for to increase the likelihood that behaviors will occur and movements will occur. And so Huntington's is thought to maybe relate to an excess of activity in that direct pathway. And so it's this sort of competition between the two pathways that's thought to lean a person's basal ganglia more towards one movement disorder or another, Parkinson's being a general paucity of movement, Huntington's being sort of an excess of movement, and hence the reason those two disorders are classified as movement disorders. However, in reality, Parkinson's and Huntington's are associated with many other structures outside the basal ganglia. So if we look at Parkinson's on the left, Huntington's on the right, beyond the basal ganglia, Parkinson's involves many structures in the brainstem, and it involves many structures elsewhere, particularly the neocortex. Huntington's also involves the neocortex, and it involves several of these deeper subcortical structures as well. So the thalamus, hippocampus, and even the cerebellum are afflicted. Parkinson's affects elements of the autonomic nervous system, particularly the enteric or so-called gut nervous system. And then over on the right, Huntington's affects the musculoskeletal system and even the heart, the cardiac muscle of the heart. And both of the disorders are associated with defects in other systems, such as the circulatory system, with recent studies showing that white blood cell function is affected in both. So these are really widespread disorders affecting multiple neurons and structures. So we have to somehow account for that and look beyond just the basal ganglia. So leaving location aside, neurologists also love to look at pathology. So the pathology in Parkinson's and Huntington's is thought to be neurodegenerative, and it's seen as a pathology, keyword being pathology. Parkinson's is linked to environmental factors, the strongest of which would be insecticides, herbicides, and head trauma. Huntington's very strongly linked, as we all know, to genetic factors, particularly a CAG trinucleotide repeat expansion, which codes for the abnormal Huntington protein, and that protein is involved in an array of transcriptional, translational, metabolic, mitochondrial processes. So it's not really sure how these things ultimately lead to neurodegeneration, but various disruptive mechanisms have been postulated. But basically, the neurodegeneration itself is seen as the pathology and the thing we have to treat. And I'd like to show you that that is probably not true. Mitochondria dysfunction is the thing that we really need to treat. That's the true pathology. So we know that Parkinson's and Huntington's involve striking defects in mitochondria. That's not really debatable, which links all of the factors I just described, environmental and genetic, actually, and can be recognized as the pathology underlying neurodegeneration. And very importantly here, and this is a flip in the way of thinking about it, that maybe not everyone will be able to make right away. Neurodegeneration, I see that as a response to the true pathology, which is the mitochondria dysfunction. So the mitochondria dysfunction results in a number of metabolic problems, particularly energy shortage, increasing levels of oxidative damage, and that is followed by a neurodegenerative response as the cell is trying to and failing to adapt to the mitochondria dysfunction. So how does that look? So Parkinson's and Huntington's, we can see as still, the former largely being triggered by environmental factors, Huntington's by the genetic factor. And both of those things, environmental factors, the ones that affect Parkinson's, they are known to induce mitochondria damage and dysfunction. And like I said before, the Huntington protein is involved in a vast array of metabolic processes, including the mitochondria. And so that can conceivably result in high levels of that can result over time in mitochondria dysfunction. So both of these things are known to, to be potentially damaging to mitochondria, and that is followed by the neurodegeneration. So if this is the correct picture, which in my view it is, then really treating the mitochondria dysfunction is the key, not the neurodegeneration, because the latter is a response. And note that this sort of, this doesn't go against the view that Huntington's, for example, has a large genetic basis, Parkinson's has a large environmental basis, and so on. So let's put all these slides together and look at this view in one slide, if you still haven't understood what I'm trying to say yet. So rather than viewing these things as focal pathologies, basically as basal ganglia neurodegenerative disorders, Parkinson's and Huntington's can be recognized as widespread mitochondria disorders, which would of course explain the involvement of multiple structures throughout the brain and body. And also it would frame neurodegeneration as a response. So I like icebergs. So let's look at this from an iceberg point of view. So it's very clear when we see a patient with one of these two disorders that there are motor symptoms such as tremor in Parkinson's, such as chorea in Huntington's. If you look a little deeper, you will see the non-motor symptoms. So mood disorders, sleep disorders, and so on in Parkinson's, cognitive and neuropsychiatric problems in Huntington's. If you go just beneath the water, so you're starting to pierce the water, yes, there is focal pathology, basal ganglia neurodegeneration. The bulk of studies and research, I would say, is focusing at trying to understand or treat the iceberg at this level. But if you go deeper, I think a number of researchers are starting to realize more and more that Parkinson's and Huntington's involve widespread neurons throughout the brain and other structures throughout the body. And there are maybe just a few thinking this deeply, I would say, viewing neurodegeneration not as the primary pathology, but as a response to the primary pathology. It's a metabolic response where the neurons are failing to adapt to mitochondrial dysfunction. That's the true pathology, in my opinion. And of that, of course, explains the multitude of environmental links to Parkinson's, and it can be explained by the genetic anomalies in Huntington's. Now, if this iceberg is true, then really all of these things are effects or responses that occur as the mitochondrial dysfunction, the impact of that trickles up the iceberg. And what we really need, therefore, are therapies at the bottom of the iceberg to treat the true pathology, the mitochondrial dysfunction in particular. Because if we're just trying to treat the responses, we're only going to have limited success. So by trying to treat the bottom of the iceberg, we can hopefully induce a similar trickle-up effect of a therapeutic response and actually maybe improve all these other things as well. So I'd like to state that that's the way Parkinson's and Huntington's will be viewed for this talk. And if you find that challenging, then well, the rest might be a little challenging as well, but we'll move on. So metabolic strategies, what are those? I just define a metabolic strategy as any intervention that stimulates generation of ketones. Generally, the primary blood ketone, beta hydroxybutyrate, or BHB, is what I'll talk about when I say the word ketone. And these are molecules that serve as bioenergetic alternatives to glucose. However, metabolic strategies orchestrate a variety of other metabolic alterations in the brain and body, and those are the more important aspect of the term. So the most common and effective metabolic strategies are, without a doubt, fasting and ketogenic diets. Fasting is just a voluntary restriction from food and or drink for at least 12 hours. A ketogenic diet is a high-fat, moderate protein, low carbohydrate diet. Basically, both of them make the brain and body run on high levels of fat. Now, the major difference between these two things is that fasting, the fat is coming in from an intrinsic source, so from the body, and ketogenic diets, the fat is coming from an extrinsic source, from the diet. Either way, large level amounts of fat are released into the blood, and the liver is the main organ. It's not the only one, but it's the main one that converts that excess fat into these things that we call ketones. And brain and muscle in particular are very adept at utilizing ketones as an alternative to glucose. And in fact, they're superior, as we'll see on the next slide. So again, fasting and keto diets have basically the same aim, which is to sort of fat adapt the brain and muscle and rest of the body to a lesser extent. And the major difference between them is simply where the fat is coming from. Now, ketones can also be given directly, but I do not call these things metabolic strategies. These are things like the medium-chain triglyceride oil. These are things like artificial ketone esters that they do elevate ketone levels in the blood, there's no question. However, they do not induce the variety of other metabolic alterations in the brain and body. And for that reason, although these can be considered ketogenic supplements, they are not metabolic strategies. Now, regardless of whether you use fasting or ketogenic diets or the ketone supplements, excuse me, there are specific ketone advantages. So compared with glucose, BHB displays a number of advantages which may be useful in Parkinson's and Huntington's. And the first one is that obviously it provides an alternative fuel source for the brain. So with Parkinson's and Huntington's, we know there's impaired brain glucose metabolism. We know there's a degree of brain insulin resistance. And so by providing an alternative fuel when glucose isn't being metabolized very well, there's going to be an obvious benefit there, and that the cell is able to utilize an alternative fuel in case the first one's not working so well. Beyond that, the ketones generate more ATP energy per unit of oxygen. For those biochemistry aficionados, you can go ahead and look up why this is, but it basically involves a significantly increased flux of intermediates through the TCA cycle. Ketones generate fewer reactive oxygen species per unit of oxygen at the same time. So therefore, they are not only producing more energy per unit oxygen, they are actually a so-called cleaner fuel per unit oxygen, producing fewer damaging oxygen species. And of course, ketones have multiple signaling functions that control oxidative stress and inflammation. So for example, they inhibit histone deacetylases and NLRP3 inflammasome activity. And these are things that it is generally thought of as a good thing to limit their effects, because by doing so, one can potentially reduce oxidative stress and inflammation. So given that Parkinson's and Huntington's show, amongst other things, impaired brain glucose metabolism, neuron oxidative stress, and neural inflammation, you can see why BHB may indeed be helpful. However, that all being said, BHB, the ketones, are still the minor contribution. They might be the major contribution of a ketone supplement, no question, but they are the minor contribution, in my opinion, of a metabolic strategy. So a metabolic strategy like fasting or ketogenic diet has widespread metabolic advantages. So in this perspective, ketones are simply another tip of the iceberg, another iceberg analogy, I am sorry, with respect to the potential benefits of metabolic strategies, particularly if we see these disorders as widespread mitochondria disorders. For example, fasting and ketogenic diets upregulate AMP kinase, which is a master nutrient and energy sensor that when it's elevated, it does many things, but it induces autophagy, including mitophagy, which is cell recycling of damaged proteins and mitochondria in the case of mitophagy. And this is useful because the cells have to recycle their mitochondria pool and remove the old damaged mitochondria to maintain their energy efficiency. The ketogenic, metabolic strategies upregulate sirtuins, which enhance mitochondria efficiency, amongst other things. Metabolic strategies upregulate PGC1 alpha, a master regulator of mitochondria metabolism, which induces mitochondria biogenesis, amongst other things. And metabolic strategies upregulate a number of antioxidants, which lessen mitochondria damage. And there are a number there that I really haven't shown. So really, it's not really, the ketones are a minor part of a metabolic strategy. The primary therapeutic potential for them lies in, as you can see, restoring widespread mitochondrial function. And if we're talking about disorders where we're viewing mitochondria dysfunction as the primary or true pathology, with neurodegeneration as a response, and this is occurring throughout the body, you can see why fasting and ketogenic diets might be very desirable ways to treat this, seeing that they restore mitochondrial function and they have the potential to do so throughout the brain and body. Okay, so let's look at some evidence. Metabolic strategies in Parkinson's. There's, of course, a major scarcity of evidence. Now, I focus on interventional trials in humans. There are many animal studies. I'm not going to talk about them. I pretty much like to look at evidence involving people largely. So, and observational studies, I'm not going to talk about them either. So there are really only 3 studies that meet these criteria for being studies that are interventional involving humans. And I will briefly discuss each of these trials. So the first one, done by the Van Italy team in New York, 17 years ago now, single-arm clinical trial involving 7 Parkinson's patients, and the trial lasted 4 weeks. The intervention was a hyperketogenic diet. This is a very powerful ketogenic diet. It was 80% fat by weight, and it was very low in protein content, which of course induces a confounder when we're talking about Parkinson's, as low protein would mean enhanced levodopa absorption compared to someone on a higher protein diet. Another confounder is that they did not have a control group. So this is very important, as we all know, to deal with something called the placebo effect. You need to have a control group to sort of tease out how much of any measured benefit is due to the expectation of benefit by the patients themselves. And so the lack of control group was another severe confounder or limitation here. And the team acknowledged both of these things. So the assessments were done by unblinded movement disorder specialists, and the primary outcome changes were changes in motor symptoms using the clinically validated UPDRS scale. Of the 7 patients commencing, 5 people completed the 4 weeks, and the average improvement in motor symptoms was 43%. That sounds pretty good. However, we do have to remember those 2 severe limitations. So I'm sure that part of that, at least, was due to the low protein content, and part of it would have also been due to the expectation of benefit or placebo effect, which, as we know, is quite powerful in Parkinson's, and it's along the lines of 20%. So it's conceivable that the majority of that benefit was actually due to the low protein content and placebo effect. But if you do want to look at the improvements per patient, here they are. So improvement from baseline as a percentage. The patient that improved the least, patient 1, improved by 20%, and patient 5 on the right improved by 80%, average 43%. So quite encouraging, but again, there were some limitations there. So our group did the next study of a metabolic strategy in Parkinson's, which is another ketogenic diet, a slightly different one though, and that was only 3 or 4 years ago. We did a randomized controlled trial involving 47 patients over 8 weeks. We wanted to double the time, and we really wanted to try and eradicate those 2 limitations from the Van Italy study. And I'll show you how we tried to do that. So we used a modified ketogenic diet, which was 65% fat by intake. That's sort of a moderately strong ketogenic diet, not as strong as the Van Italy one. And our control was a healthy recommended New Zealand healthy eating guidelines, low-fat diet, lots of vegetables, and so on. And it was 15% fat by weight. So quite a huge disparity in the fat intake, as you can see. And very importantly, we had normal protein content in both diets, about 1 gram per kilogram, which we controlled in both arms to the best of our ability. And that would be for an elderly population of Parkinson's seen as normal or even slightly high protein levels. For someone doing weightlifting and they're 30 years old, maybe that would be a little on the low side, but it's all relative. So for these guys, that's absolutely not a low protein diet. And very importantly, we equally promoted both diets as potentially beneficial. So we knew there would be a placebo effect applied to both arms of the diet. You can't really avoid it. So what we've tried to do was make sure it was applied equally to both arms. And I think we succeeded in that. For assessments, we used blinded MDS certified neurologists, and we ensured that the patients took their medications at the same time prior to each visit. So we avoided any confounding there. And they were assessed by the same neurologist, same weekday, same time of day at every visit. And that's very important because Parkinson's can fluctuate even from hour to hour, as many of you know. The primary outcomes we were interested in were the changes in non-motor and motor symptoms and motor complications. So basically Parkinson's in its entirety, and we used the entire MDS UPDRS, a more updated version of the UPDRS, to do that. So what did we find? 47 patients entered the trial, but 3 dropped out before even starting the diets. So we had 44 start the diets, 38 completers, pretty similar in each group. Long story short, the keto group improved non-motor symptoms by 41% compared with 11% in the low-fat group. So looking at the graph, the blue line is the low-fat group, down is good. They improved by 11% from baseline by week 8, but the keto group improved by a substantially larger, clinically and statistically significant 41%. That was a pretty profound difference. And interestingly, the top 5 most improved symptoms were those that tend to be, as a group, fairly levodopa unresponsive. So urinary problems, pain, fatigue, daytime sleepiness, and cognitive impairment were the top 5. There were others, just listed the top 5. And this is really encouraging. It means that maybe an optimal strategy for someone with Parkinson's would be to use the traditional medication-based strategy plus a metabolic strategy, such as a ketogenic diet. Now, in both groups, I should say the motor symptoms also improved by about 20%, but we didn't get too excited about this because basically there was no between group difference. And we know this was at least partly due to a healthier overall diet. For example, neither of these diets had beer and ice cream and things like that, which, you know, a lot of these patients were consuming considerable amounts of before the trial. So both diets were probably healthier than what they were on before. And of course, the placebo effect would have applied to both arms of the trial, albeit equally. And that would have been, that would explain at least part of that improvement. So, but if you want to look at the score changes, so again, improvement from baseline as a percentage, the non-motor symptoms, red is keto, blue is low fat. As I said before, keto group, that's a statistically significant and clinically significant difference. That was a remarkable difference. The motor symptoms in the middle, there was really no change between the two. And the motor complications on the right, again, there was no change. Although it looks like keto, the red bar, is a little higher than the blue bar, the low-fat diet. Yes, it did seem to get, it did seem to improve, but when you did the statistics, it did not quite make it there. So we can't comment on it. So again, more encouraging findings from that second study. The third study was done by the Kerkorian team over in Cincinnati, and they published their findings a year after ours, and it was a randomized controlled trial too, involving 18 Parkinson's patients, and they really looked at Parkinson's patients with mild cognitive impairment, or MCI. So they were focusing on cognition. Their intervention was a low-carb diet, which could be thought of as a weekly ketogenic diet. It was 42% fat by weight, and their control was a low-fat diet, kind of similar to ours, I guess. It was a healthy low-fat diet, lots of vegetables and so on, 24% fat by weight. I couldn't find any mention of assessors in the paper, so if it's there, I apologize to the authors, I just couldn't find it. And their primary outcomes of interest were basically cognitive and motor. So they looked at changes in lexical access and memory using a number of cognitive tests, and motor symptoms using the motor component of the MDS UPDRS. So the Kerkorian team had 14 completers out of their 18 patients, which is pretty good. And basically, long story short, low carb improved cognition compared to low fat. So if you look at their findings, they're looking at word association on the top and paired associate learning on the bottom graph. And you can see in both cases, as shown by the green box, that the low carb group improved compared to the low fat group, statistically significant. And their motor symptoms didn't show any between group differences. So again, fairly encouraging. All right, what about Huntington's? So unlike Parkinson's, there are no interventional human-based studies in Huntington's. Again, there are animal studies, but I'm not going to talk about them. We are currently running a single-person case study. This would, I guess, be the first case to my knowledge in the world of a person with Huntington's on a metabolic strategy. And, you know, at some point when conditions permit, with respect to the COVID restrictions and so on, we'd like to do a larger study, but right now it's not the best time. So we'll briefly discuss the results to date. Remember, this is N of one patient only. So, you know, it's extremely preliminary. But anyways, this is a case study, one patient, 1 year of a metabolic strategy. We're almost done the year, actually. And this was a 41-year-old man with clinically and genetically confirmed Huntington's, 47 repeats, who was experiencing an ongoing deterioration in basically all four main components of Huntington's: chorea, cognition, function, and behavior. We'd been seeing him in the clinic for about a year prior to starting this, and the decline was visible. The decline was visible to his fiance, and basically both he and her were extremely concerned about this. And despite everything that had been tried, it just seemed as though the Huntington's was progressing. So I didn't want to start a metabolic strategy, but they sort of knew that our department was involved in these things, and they were quite desperate. And so I thought, well, I didn't want to start one in him because he was at least moderate severity at the time, and he was progressing fairly rapidly, and he also had a very low body mass index and so on. But we said, oh, well, let's try it anyways. So the intervention in this case, we changed it a little bit for him. It was a time-restricted ketogenic diet. So we combined fasting and ketogenic diet in this metabolic strategy. So combined metabolic strategy involving 2 meals a day, no snacks, 60% fat by weight. So relatively short fast, but it ensured that we gave the body lots of time, 22 hours every day, where there was no food coming in. And when he did eat, it was a sort of mild to moderate ketogenic diet. Blinded neurologist and neuropsychologist did all the assessments, and our primary outcomes, of course, are the ones that he was declining in. So motor symptoms, cognition, function, composite score, quality of life, and problem behaviors. The composite score is, as some people will know, it's a combination of motor, cognition, and function symptoms. And it's a very robust sort of overall picture of how someone with Huntington's is doing. Okay, so the timeline was and is as follows. Basically, we started this strategy in April of 2021. We did the 6-month or week 24 assessment in September of 2021, and we plan to do the 1 year or week 48 assessment, which is almost 1 year, in March of 2022, which is next month. So I can only give you the 6-month data, but I can say that we have measured improvements in motor symptoms, function, and the composite score. So if we look at that on a graph, blue is baseline, orange is 6 months or week 24. Motor symptoms improved by 27%, so down is better in that case. And function improved by 48%, down is better. And the composite score, interestingly, improved by 15%, up is better. That's not a huge jump, 15%, but considering this man was visibly deteriorating very quickly, and then, you know, he's not only stabilized, but perhaps gained a little ground back after 6 months of this approach, I think that's encouraging. Remember, it's just one patient. If we look at problem behaviors, he also improved in, he had 4 main problem behaviors at the start of this: anger, apathy, disorientation, and irritability. And if we look at that graph, again, blue is baseline, orange is 6 months or week 24, we can see that all 4 of those things actually improved. Down is better. So that's highly encouraging. The one thing that got worse was anxiety. He didn't have any measurable anxiety at the start of the case study, and he did have measurable anxiety at the 6-month or week 24 mark. There may be other reasons for that. I don't think I need to get into them for this talk. But basically, let's wait and see how we go. Remember, this is incredibly preliminary data, and we're just sort of seeing what happens in Huntington's. And yeah, one person only. So it's interesting, but extremely preliminary. And he's had no weight loss, I should mention too, which is great considering his very low body mass index. Okay, so in conclusion, thank you for bearing with me the last half hour. I would state that rather than maintaining the current concept of Parkinson's and Huntington's as focal pathologies, that is basal ganglia neurodegenerative disorders, it's better to recognize them for what they are, which is widespread mitochondria disorders, whereby they afflict multiple structures in the brain and body, and the neurodegeneration is a response to the true pathology of mitochondria dysfunction. Now, there's a lot of excitement about the specific advantages of ketones. They do exist, but in my opinion, they are just the tip of the iceberg with respect to the primary therapeutic potential of metabolic strategies, which is in restoring widespread mitochondria function. That's the A game when it comes to these things. In Parkinson's, there's preliminary evidence suggesting keto diets may improve non-motor symptoms. Unfortunately, we lack even preliminary evidence in Huntington's. So thank you very much for watching my talk, and hopefully I am now there to answer any questions.